All right, welcome back, everybody, to session number 2 of our investigation design essentials. So just a reminder, the purpose of this 2-part series is to help you guys think like a scientist and do what scientists do, right? But again, most of you are probably watching this so that you can better prepare for that AP test, right? Maybe some of the multiple choice questions, but definitely that first free response question on the second part of the AP test. All right. And some things you might be asked to do with investigation design. You might be asked to analyze and evaluate an investigation that's already been written up, and you're just kind of looking at the data and the methods, right? Or you might even be asked to design your own investigation, in which case the content of these videos will be helpful either way. So if you would, in video number 1, you would know that we talked about independent variables, dependent variables, and control groups in that first video, right? So definitely worthwhile to make sure you go back and review those terms, because these are both videos, while split into 2, are essentially one cohesive topic. So you definitely need to have a solid understanding of these 3 terms and what they mean and how they apply to investigation design and why they're important. All right. But in this particular video, we're going to talk about validity, which is essentially having controlled variables, reliability, which means the results of your investigation would be reproducible and getting the same result. And then how can you look at some data and draw some correlations, draw some conclusions, identify trends, etc. That is going to be the primary focus of this video. So a reminder, in video number 1, the context we used was testing different soil amendments on tomato plants, right? And so the investigative question or the testable question that I generated for you guys was, which soil amendment, organic fertilizer versus chicken manure versus kitchen compost, will cause the greatest increase in tomato yield in my garden? So my independent variable, right, is the different soil amendments, the fertilizer, the chicken manure, and the kitchen compost, right? Going to be applying those to my garden soil to see which one produces the greatest increase in tomato yield, right? So my dependent variable or responding variable would be the tomato yield. All right. And so we ended last video right around here where we just talked about a summary of how we do things, right? So we had a testable question. We have a control group, which is an unaltered sample, so a group of, in this case, a group of tomato plants that we don't add anything to the soil, right? Then we have our experimental group, which has an independent variable change to their system. We observe the results of all of them, right? Then we measure that difference as a dependent variable. Okay, moving forward then, we're going to talk about validity, right? So a valid experiment, okay, is one in which we have as many controlled variables as we can. And there is not a magic number of controlled variables, because the number of controlled variables that you ought to have really depends on the system that it is that we're testing, right? So again, within the context of this investigation, right? So tomato plants, that's how we're going to kind of like think about things for now. But we talked last time about having only one type of change to the system, right? So that is your independent variable or manipulated variable, synonym, right? Only one type of change to the system is to be tested, okay? All other potential impacts must be controlled. Okay, so what does that mean? So so anything that might increase or decrease the growth of my tomato plants or my production of tomatoes in my garden, right? all other things that might impact that production need to be kept the same. So all of my tomato plants would need to receive the same amount of water, right? They would all need to be in the same, probably the same general location so that they're getting the same amount of sunlight, right? The types of tomato plants that I'm using, how do I go about controlling weeds and pests, right? All these things that might impact the growth of my tomato plants, I need to keep those the same, right? All of them kept the same, all right? And as much as I possibly can. Now, because the purpose behind that is I want to see how the soil amendment and only the soil amendments are impacting my tomatoes. Right? If I want to be a scientist here, I can't just throw a bunch of different changes at the system, right? Because then I can't identify which of those changes caused the results that I was looking for, right? I need to isolate a single change, right? And keep all other things constant. I don't know how better to say that at this point. All right? So we need to have a valid experiment by controlling as many variables as we can. All right? Now, what you might see in your AP test, right? They might ask questions about what could you do to make this experiment more valid? In which case you would look at the information given and you'd say, oh, control X or control this variable a little bit better. Make sure they're all getting the same amount as that. That's probably how the validity measures is going to appear if you see something about validity on the AP test. All right. Now, if you are writing a procedure, right, the controls, right, so if you're writing a lab report, for example, right, in your class, or if you're writing up designing an investigation on the AP test, right, these controls need to be explicit in your procedure, right? So if I was writing up a procedure for my tomato plant experiment, I would say all samples will receive the same amount of water. All samples will be exposed to the same amount of sunlight. All samples will be weeded once per week, right? Etc. I'm going to make those explicit in my procedure. Don't just imply them. Okay? Now, the other term we want to look at is reliability, right? And it's related to validity because a valid experiment should potentially lead to a reliable experiment. But what we want is something that's reproducible, right? Which means that if someone else were to run it, or if I were to run the experiment again, right, I would get the same results. So, but first and foremost, we want to be able to predict reliability, meaning that we have a large enough sample size. Okay? So, first of all, sample size and reliability are not necessarily the same thing, but sample size is important for us to assume or predict that an experiment is reliable. So, in this example, again, if I were growing tomato plants, right, I would not just have one of each, one control and then one in chicken manure and then one in fertilizer and one in compost, right? Having a single plant is not a large enough sample size because if you've ever grown any sort of plant, right, you would understand that there's a random variation, right? Okay, they're not like necessarily genetically identical. Okay, so one tomato plant might produce more than another one, even if everything is kept the same, right? So we needed a large enough sample size in each condition to rule out random variation, right? Now, if, right, let's say you've got 3 plants or 10 plants in each condition, right, and you consistently see a change in results, then you can predict, oh, this is, I have a fair degree of confidence that this type of soil amendment produces the best results. If all you have is a single plant, you don't have much confidence in those results. It could have just been random variation that produced that result. Okay? That's why I put that first. But sample size is not the same as reliability. Okay? But we do need a large enough sample size in the initial experiment to assume or predict that these results are reliable. Now, Second then, right, in order to consider an experiment reliable, right, it would need to be run again, right? It could be run by somebody else. It could be run again by you, right? But we need to have another running of that experiment, okay? And if it is run again and we get similar results, right? We run that experiment again, we get similar results, we would then say that that is a reliable, or the results of that experiment are reliable, which means, hey, we have good, high confidence, right, that somebody else might get the same results, which means we would have high confidence in how the independent variable impacts the responding variable. Okay, moving forward. So, analyzing data and drawing conclusions. So let's say we set up our experiment. We have our test plots of our control group, and then we've got our chicken manure plants, and we've got our plants in organic fertilizer and our plants in kitchen compost. We've got all that set up, right? We give them the same amount of water. We give them this, basically everything's the same with the exception of the soil amendment, right? How then do we go about analyzing and interpreting the data or drawing conclusions? So one of the things that you will not be asked to do, you will not be asked to just make up data, right? You will not be asked to just make up data. Okay? Now, you might be asked to predict a trend in data, right? And so they might say, if we made this change, how do you think the responding variable or dependent variable will change? In which case you would probably say, I would predict it will increase or decrease or stay the same. Generally, they wouldn't ask you to calculate that unless you could follow some sort of pre-existing trend line or something. Okay? So, might be asked to predict a trend in data. That's not that uncommon on the AP test. Likely, what they're going to ask you to do is to identify a trend or a correlation. Okay? Now, the way they might look is they might ask you to identify something as being positively correlated or negatively correlated or potentially having no correlation whatsoever. So you'd look at the data, right? Is my slope, if you put it on a graph, is the slope positive, right? We would say we have a positive correlation. If the slope's negative, we'd say we have a negative correlation. Now, you're not going to have to do statistical analysis and identify like an R or R squared value or run those types of things. So those of you who've had statistics on the AP environmental science test, it is not expected for you guys to do those statistical analyses. There's no standard deviation calculations or anything like that, okay? But what you might ask to do quantitatively is calculate a percent increase in something. That's a really common thing that you might see on the AP test, right? Or often identify what type of correlation, okay? Or you might just be asked, hey, what's the relationship between this and this? Those types of questions could be asked in a lot of different ways, whether they want you to actually give a numerical response that will be explicit in terms of, they'll say, calculate or something like that, okay? Now, so let's go through and take our tomato plant investigation to the end. So we produced some data, and I made the choice of putting 3 plants, all right, which would be our minimum. All right, so I don't have that big of a yard, so I couldn't grow more than 12 tomato plants. My garden's not huge. All right, so I put 3 plants in each condition. I got 3 control plants, 3 plants in the chicken manure, 3 in the organic fertilizer, and 3 in the kitchen compost. Right? Then, at the end of my tomato growing season, right, I tally up the total amounts for each plant, right, add those up, divide, and I get my average. I'm going to assume you guys know how to do averages. Right? Do the same thing for chicken manure, same thing for organic fertilizer, etc. All right? So, now, when you see averages, that's typically what you're going to use for your calculations, if you were to make calculations like a percent increase and so on. But it's definitely still worth your time, right, to look at the individual data. right? So yes, the average is higher in some circumstances, but how consistently are the individual plants higher, right? That's just adding to your interpretation, okay? But for the most part, we're going to focus on these average amounts, right? So what can I draw? What conclusions can I make? Well, control group in all cases was the lowest. So I could say, while all soil amendments showed an increase in average yield, right? Chicken manure was clearly the highest, provided the greatest increase in tomato yield, with an average increase of 0.85 kilograms per plant, or about a 71% increase compared to just 9% for organic fertilizer and 26% increase for kitchen compost, right? So again, we have this control group, and what we're going to do is we're going to compare each sample back to what we see in that control group, right? That's a typical and well-established way of comparing things. Now, we can of course compare between the other samples as well, right? But we definitely want to see, does this produce every desired result, right? So it might be that at the end of this, I'm trying to decide what's best. Well, you know what? Maybe this is a big enough increase right here that I'm happy with, and this way I don't have to go buy chicken manure or buy organic fertilizer, right? So there might be a cost comparison that I make at the end and stuff like that. You won't have to do that on the AP test. I'm just saying, in general, in life, if we're going to put these things to use in general life, it's good to think about how we might then use these results. Just because a particular variable gives the best results doesn't mean that's the one we're going to go with, right? So anyways, this is the type of thing we want to be doing. Now, if you did do a calculation, right? So any numerical response that you calculate, always, whether they ask you to or not, always show that calculation. And of course, if they ask you to calculate something, you have to show your work to receive credit, right? But if you make a calculation, right, as a part of a response, still show your work even if it didn't ask you to make that calculation. Always, always, always show the work in your calculations. All right, I am going to cut us off there. I hope you guys do great on your upcoming AP tests.